Okay, the webinar has started.
Yes, I'm just waiting for people to join, at least 10 or something.
Yeah, no problem.
Yeah.
Yeah, we can wait like for another 5 or 10 minutes.
Yes, we can. Yeah.
It doesn't bother me.
Okay, good afternoon, everyone. My name is Lihle Kulu. I am the Deputy Secretary of the Society of Neurosciences of Africa, and I'm very excited today to be introducing my former colleague, neuroscientist friend, all the way from Morocco, to our fourth webinar of the year. And I'm going to actually allow my other colleague, Neza, who is the representative of SONA in Northern Africa, to do the formal introduction. But before I do the intro, I hand over to Neza. I also just want to congratulate Professor Abosi, who has moved from being Dr. Abosi to being Professor Abosi. A hearty congratulations to you. And I know that you are such a hard worker with a big, beautiful, big lab. I'm very happy for you, and I wish you the best in this new position. Okay, I will hand over to Neza now. And all the best. I'm excited for this talk, Walid. I can't wait to hear what you have to say to us. Thank you.
Thank you, Lily. Thank you. And I also welcome everybody to this fourth SONA webinar. Today, we delve into the fascinating evolution of biostatistics with our distinguished speaker, Professor Walid Aboussi. So Professor Aboussi is a neuroscientist with a master's in bioengineering and a PhD in neuropharmacology. He completed postdoctoral research at the University of KwaZulu-Natal, focusing on neuroinflammatory and epigenetic changes in drug abuse models. He then managed the Behavioral Neuroscience Core Facility at the University of Dundee, UK. Currently, as Lile said, he is a professor of neuroscience at the University Mohammed V in Rabat, Morocco. We are colleagues, actually. And we hope this webinar deepens your understanding of biostatistics. So please join the question and answer session after Dr. Boussi's presentation by writing your question in the question and answer space. So I wish you all an instructive webinar, and Professor Aboussi, the floor is yours.
Thank you, Nizha. Thank you, Dishli. The pleasure is mine, actually, to attend this webinar and to be presenting one of the tools that we use usually in our studies. Welcome to anyone interested in statistics or biostatistics. So what I'm gonna do today is just actually the title is From Fossil to Life and Navigating the Evolution of Biostatistics, but mainly I will be focusing on the why it is interesting to use the R software to analyze data, to generate graphics, instead of using classical softwares. So, concerning the evolution of biostatistics, so early beginnings and the biostatistic has its roots in the study of natural phenomena, including the analysis of fossil records. So early biostatisticians relied on manual calculation rudimentary tools to interpret data about past life forms, about life generally. But by the advent of computers, everything has been revolutionized. So now we start using Excel as a start, which it enable more sophisticated analysis. And since then, other classical statistical softwares that I'm sure most of you, or at least some of you, have been using, such as SPSS, GraphPad Prism, STAT, or SAS, become essential for our modern data analysis. However, with time and with the new techniques like all the techniques that we are using now, like the omics, genomics, proteomics, whatever electrophysiology data, all these data are big data. So when using this classical model like SPSS, SAS, or Stata, sometimes we may face some limitation. So generally, the most known limitation with these softwares like SAS and SPSS or GraphPad Prism, that they are closed source software. It means that you have to buy a license for using it. And the cost of this license is very expensive that individuals or small labs generally cannot afford. And these softwares lacks most of features in graphical visualization. So actually, when we want to express our data, sometimes we need to express a lot of components at once. Sometimes we need to express them in a way that answer our research question. And when you have like softwares that cannot be modified, they are closed, then you cannot express what you have in mind. So these software generally, they lack features in graphical visualization. Then there is the R tool. R tool actually is totally different because R tool is programming. So actually the limit for R tool when analyzing data is your imagination, because all what you want to do, you write it as a script and it can do the analysis for you. And the language that are used generally is like an easy language, because one of the oldest programming languages. The scripts are short, and generally they are already available in form of packages that we're gonna go through later in this presentation. Other thing are, because it's an open source, it's for free, its installation, it's easy, there is no hassle with it at all. And if we look to recently published data, we can see that people are generally, most of the people or most of researchers and in our field, they are transiting from classical softwares like TransFlow, Stata, SAS, SPSS to GraphPad Prism to R. So R actually is the most used software by most of researchers. That means actually that the community that is using R is growing. And if I say that the community that is using R is growing, that people that are developing script to analyze data mostly is going to be enriched. That's why it becomes time to time, R is most be used, and people, they're looking for it to use it to analyze their data. And either ways, qualitative or quantitative data, R is sufficient actually to analyze these data. So, and we can see that like in the last years, like a lot of software being used, and we can see after some point with most of software, the SaaS data, there is a decline. But if we look to R, it's always ascending, it's always exponential, because people use it, people develop scripts for it, and then it became more easier to use, it became more sophisticated, it became more capable in performing data. So one of the things that making people actually look to work with R is its capability of performing what we call data wrangling. So most of the classical softwares, like, as I said, SAS, SPSS, or GraphPad Prism, actually all what you can do is just like classical analysis. You can look to normality, you can do some transformation, normalization, then you pass to comparison using, I don't know, parametric tests or non-parametric tests. But the hardest thing when dealing with big data, it's discovering the data, trying to explore the data, trying to structure the data, to clean the data if there is outliners, to enrich the data if the database each class there is missing values. So you have to add some functionality, some correction to add the missing values, to validate the data, and then to publish the data as graphics, as visualization that's going to explain your data. And all these steps are what we call steps of data wrangling. It's impossible to do with the classical method, but it's easy to do it with R software. So what is R? Actually, R is a programming language specifically designed to do statistical, has to do its unparalleled flexibility and have customization capabilities. So unlike the old software, which often come with predefined procedures, so we have just to click on buttons generally to execute function. R is a programming language specifically designed for statistical computing and graphics. This allows users to tailor their analysis to their specific need, to their specific research question, and then to create custom function and packages. And remember that word of packages, because R, it's mostly function with packages, and we're gonna go and talk about what are these packages and how we can use it for all kind of analysis. So R is use packages, and that means it helps actually to integrate like novel data, complex data, whatever kind of data, if it's numerical or it's integer data, it can implicated, it can be analyzed by our software. So what is this packages? So actually, R, as I said, it works with packages. So usually when we are programming with R, actually there is like some function that's already prepared. And this function that they are already prepared, we call them packages. So before starting the analyze, usually we use what we call the pre-modeling stage packages, and that we can use a ggplot, for example, for data visualization. We can use packages to transform data to log or exponential data or to normalize data. Then we can use other packages to add the missing value or to amputate other values, other packages to detect outliers. And there is many packages according to the type of analysis that you want to do. Then after pre-modeling stage, there is the modeling stage. And this modeling stage, here we can do like proper statistical analysis. We can do correlation, regression, we can do classification, we can do comparisons, we can do clustering, we can do time series, and we can do survival. And for each function, there is a package that is proper to it that can be used. And after that, once we finish, it's come this stage of validation. So usually with R, there is the pre-modeling stage, just to analyze the raw data and try to clean these down raw data. Then this modeling state to look for its normality, its repartition, and everything, and start doing the statistical analysis, and then validating these analysis. And for post-modeling stage, there is packages like LS means for general model validation, regtest for regression validation, binom tools for classification validation, and etc. Right. So unlike old softwares, as I said, R and R Studio are open sources. R is the language, and R Studio, like it's the graphical version that it's being used to do statistics. So it's an open software. So therefore, you can utilize this tool without any requirement for a license. You can also work towards the development of the R language by developing packages and customizing its code and through the resolution of its existing problems. So actually, to install it in your laptop, there is nothing easier than that. It took like two steps. Usually, the software on itself, it exists on a site that's called Posit Go. So all what we do is just we can go to that Posit Go, and I don't know if you can see it here, to the Posit Go, and then we're gonna find all kind of our studios that we can install. So we can install our studios on Windows, on Mac, on Linux, and it's for free and it's easy to install. All right. Then there is to work with R, we need, as I said, packages. And for packages, just in the recent year, like just while looking today, I found out that there is more than 18,000 available packages that you can use to analyze whatever type of data you have. You have genomics, epigenomics, electrophysiological data, epidemiology data, data packages to analyze simulation, data to analyze, I don't know, fluorescence for flow cytometry, whatever technique you use that generates massive data, there is a package that is available to analyze these data. And to look for these packages, all these packages are developed by Crane. So all what we do is just go to the Crane website. And if we go to the Crane website, sorry, I'm just gonna reduce this a little bit. To the Crane website, this is the page that you're gonna find. So first, you're gonna find all the mirrors for the Crane, for the R. Then we can look for the R sources, where we can find scripts and the neuralist script to do analysis. And there is the packages. And if we look to the packages, if I'm gonna look to the packages that sourced by publication, there is the number, it's over 20,000 package that can be used to access cryptographic, for example, to do the economic and production efficiency. But one of the most packages that is used by biologists, it's called, I'm just gonna look for it. Find. Okay. Evidency. So evidency in that package, actually, it has all the functionality that we can find in the classical softwares, and it allows all kind of analysis. All right, so this is current. This is the place where we can find packages. Also, with SPSS, what we can do, actually, it generates visually appealing plotting and graphing. And there are various popular libraries like ggplot2, and that library, it is usually able for aesthetic creation of graphs, and it's a friendly user application. And even the ggplot, we can find it also in the same website on Cran that allows to download, upload this package into the R Studio. All right, now what is the R environment look like? Actually, when I'm talking on R, you're gonna find it's concern, I'm just gonna open it on my laptop directly. There is three environments. There is the console, and in this console, actually, where we can put all the things we need R to do for us. So if you look at it at a first, you can work with it basically. It can do all kind of calculation. For example, I can do that, and it can do all kind of function. So I don't know, I can calculate the log of 10, for example. So I can use it as a calculator, I can do exponential, I can do whatever I want with it, and it will always generate data. But also with R, with the console in R, where I can put the function that I want R to execute, I can use variables and not just calculation and functions. So I can accommodate X, I don't know, I can put X equal 3. So I'm assigning 3 to a variable which is X. And then here, once I accommodate a variable to a number, to a calculation, actually it's gonna go to another space which called the environment. So in this environment, all the functions, the variables, the processes that I want to do is just gonna keep it in that part. In this environment, actually, I can also use it to import data sets. So after that, actually, I can do a lot of things. I can assign another variables, like I can make y, for example, equal 10. And then I can calculate this function easily, x plus y. So I encourage you, if you are interested in R, to start with doing with this simple calculation, and then to find out how it's like to feel to use small script like log, like exponential, whatever. So after that, actually, I can do vector values. That means if I want to work with big data, I can put this data into R directly. So for example, if I want to do, I don't know pharmacology, so usually I working with mice, and I'm scoring their performances in behavioral tests. So usually what I do, if the data it's not large, I just can go scores of my animals, and then I will assign to the scores of my animals. So I'm just gonna do scores, and then I can just go C to assign column if I want to do a table, and then I can just add the scores of whatever it is. So I'm just gonna do like anarchical numbers, whatever are these numbers. All right. Then these numbers, once I have these scores, it just gonna generate them. So now I have values, like I have values that is from 1 to 8 values. Then from these values, I can do whatever I want with them. I can calculate the mean, the standard deviation, or whatever. I can calculate the mean of these values. So I just have to write mean, and the mean of what? The mean of my scores. Then it's gonna calculate directly the mean of my scores. I can calculate the maximum value of my scores. I can do the minimum values. I can do whatever I want with them. So in the console part. So the console part is just a place where I can do function, write scripts, and then attend the data. And in the other part, we have here the global environment, but here we have what we call the plot the plot side. And this plot side, generally, we use it to see the graphics that we're gonna plot later. And here we can find also the environment, the folders that we have on our desktop if we want to go and look for something else. So as I said, here in the console, actually, it's the main part. We can do the vector of values. We can do a sequence of data of elements. We can also do names if we want. And just when we're working with names, usually what we do, we just have, when assigning names or not numerical parameters, what we do is just we have to add apostrophes. So I'm just like adding names. I don't know. I just can assign names like Ahmed and Mark. So at each time, I have just to add apostrophes and no, and Virgo and Sherlock. All right. And once with R, one of the drawbacks of R is what we call case sensitive. That means using capital letter or minor letter is different, is gonna affect the results. So usually we just, because it's calculated them and incorporated them differently. So then when we generate here, we have the names. Here I make kind of mistake. Download. All right. So I imagine you get the idea. Then I can do the summary of my score. So whatever function I needed, I can do it with the R. Okay. That is the R. So that is generally the environment of R. So whenever I need a package to do whatever, I have just to install the packages. And here it's depend what I want to do. There is many packages I said available. Like for me, if I'm working on doing statistics and I want to plot graphic, I can just go and plot like what we call the GG plot 2, which is a package that it allows me to plot graphics differently, and then it's going to be installed directly to the system. So it's very friendly user and it's not complicated at all. So now what I'm gonna do, we're just gonna do just to familiarize you with the software and the R and how it works. I'm just gonna use a data from our lab to show you how it does work. Usually, I don't know if you are familiar with genomic data, but genomic data usually it's look like this. So usually when you are working with transcriptomic or epigenomic, what you're generating, it's a billions of data at once. Like in this study, we are looking for specific sequences on the genes. And if we look here, you're gonna find that the data, it's enormous. So actually it's billions of the data. So we cannot manually go through all the data on all the chromosomes and all the genes and all the nucleates to look for outliers or to correct data. But with R, we can do that easily and simply. So here, these data, like if you can look here, we're already here at 540,000. So it's a 500,000 entry already. And usually when working with genomics or transcriptomics data, it's not easy to do them with the classical software like SPSS or GraphPad present. So what we do It just we write scripts, and then we use scripts on the R Studio. So usually, as I said, I can clear this terminal now, and I can use data. So I'm just gonna open a file that script that I already prepared. I'm just gonna open a file on my laptop. So it's easy, like opening any file with any other software that you are usually using. And I'm just gonna go and look for R for biologists. So I'm just gonna go for the first lesson, and here what we're gonna be doing for today. So just to give you an idea what we're gonna doing. So here we just gonna try to show you how we can download data to our software, whatever this data, because with R we can work with data that it became as CSV files in those with most of the machines that used in biology. Generally, the data that generated it's usually in CSV format. Then it also can read directly Excel format. So there is no problem usually when uploading data to the R Studio. And then we're gonna see how we can create a quick plot, how we can analyze the data, and how we can generate multiple file format for the graphics, either scatter graphics, heat maps, or whatever. It depends on the question we are asking and our objectives. So as I said, when you're starting, you have just to choose the right packages that you want to work with. So as I said, the first thing we have to do is to install the library package. Like for me, if I want to do graphics with R, I need this package. It's called the ggplot2. Here I already installed it, and if I want to look to the packages that already installed on my system, I just can go here on the graphical console and look to the packages. And here I can look to all the packages that they are installed on my computer, on my R Studio. So I already installed the ggplot2, so there is no need to install it again. But if you need to install it, as I said, there is a simple function. It just install packages. And then what you do, you just choose the name of the plots that you want to do. Like for me, here is the ggplot2. I've already have that, so I'm just not gonna do that. Then to open the files. To open the files, like for me to open my data files, my genomic files, I'm just gonna go to File and easily and open the file. I'm just gonna open it here as a CSV format. I will just check that all the data are here and then open it. This is a one way to open it. And another way to open this data is also I can open it from the environment. You can see here there is import dataset. And here I can import my dataset, either format from Excel. If I have already been working on another software like SPSS, SAS, or Stata, I can upload data from these softwares. So I can do that and just go to my laptop, like my data are on my desktop, and just go and look for them. All right, and this is my data, and now I can use it. All right, so here's my data. I can rename my data. So for me, I'm just gonna call it my data. All right, and you see here in within my data, there is no heading. So the first row of my data, it starts directly with chromosome 1. It starts in the situation of a gene on my chromosome. It starts, its end. and the type of that nucleotide. So here I'm just going to choose that there is no heading. So it's going to consider the first line as the first line. I'd go for the row names automatic. So he assigned V1, V2, V3, and V4 directly automatically because nothing assigned here. And then we can change that, and I will show you how we can change that. But the most important thing is to know how to import the data sets and to download the data. So here, now I import my data to the app. So here is my data, and it tells me that is my data. It's gonna be read from a position on my hard drive C on my desktop, and this is the folder name for biologist, and this is the data you're gonna be working with. All right, and now I can see all the data are here on my data. It's just because it's a large data, it's gonna be a bit slow to show it all at once. Then another thing here, I said because these data, I can do change their name. So I want to show the head of data and change it. So to show the head of data, it's straightforward. I just write to have to write head and add the name of my data that I have here on my environment, that the name I attributed to my data. And then I will just set enter. And you see here, it just attributed V1 to the first column, V2 the second column, V3, V4, V5, V7, V8, and V9. For me, if my interest is just in the first columns, that means the chromosome, the start, the end, and the position. So I don't need all this data. So I'm just gonna change the heading on these data. So to change the heading on these data, actually what I'm gonna do, I just gonna rename these data. So to rename these data, I just gonna check this function. So I'm just gonna names on my data from 1 to 4. That means from column 1 to column 4. So my data from 1 to 4. All right. And then C is to attribute. Always I will use C to attribute. So the column 1 is going to be called now from chromosome. V2 is going to be calling it start. V3 is going to be the stop position. And V4 is going to be the type. So either I can, from the console, I can directly click on command and enter, and it's going to execute the function directly. And then here, if I want to check if these headings are changed or not, I just have to write head my data again. And sorry, I'm just gonna delete this. My data. Again. And I'm just gonna click enter. And here we can see now the first column, now it's called chromosome. The second column, it's the starting point of my nucleotide. The third column, it's the stop point. And now is the type. Now I can use this data actually to plot a graphic. And for plotting a graphic, I have to use a function which is called the ggplot. Now I will just show you how that ggplot actually it does work. So the ggplot actually, when we're gonna go look for it. So the GG in the ggplot, it stands for the grammar of graphics. The grammar of graphics provide a more intuitive way to create and customize figure. It's just like a sentence is based around the combination of nouns, verbs, and adjectives and other elements that work together to contribute different facets to the meaning of a sentence. ggplot, it does the same thing. So first, we have the data. So data visualization are made of several key elements that work together. So the most basic structure of the data visualization, it begins with the data themselves. As I have here my variable, first variable, second, third, fourth, and the third variable. So then the various column of variables of that data set are mapped into different aesthetic properties of the figures. So for example, certain variables are mapped to the X and the Y axis, and then you have other variables, in this case, variable 3, which is mapped to identify the point color in the figure. So variable 1 is going to go to, I will assess it, or I'm gonna map it to the x-axis, variable 2 to the y-axis. For variable 3, I can choose for it a point color, and variable 4, I can choose for it something else, like I can choose for it a solid diagram or points or whatever I want. And then anytime a variable is used to describe an aesthetic or visual component of the figure, in this way, that is called mapping of variable to aesthetic properties of the plot. And finally, we can choose aesthetic mappings need to be presented using a geometric object. So this is what determines whether a figure will be a scatter plot, a box plot, line plot, or whatever other type of plot you want to create. So I hope you. So far, the three elements we've gone over include the data. So the data are the basics. Then we have the aesthetics, how we want to present the data, each variable to what we're gonna assign it. If we're gonna assign it to the x-axis or to the y-axis or to z-axis, or we're gonna give it a different color, or we're gonna give it a different structure. And there, the geometry, and it's the graphic in itself. It's gonna be a scatter, it's gonna be a pie, it's gonna be a head map, or whatever. So generally, and we can add other graphical elements like the coordinates or the theme to the graphic, how we want the graphic to appear. So now let's just do a quick overview of the anatomy of using ggplot, or the syntax of how ggplot functions come together. So first, you begin with the ggplot function. Now note that the package is called ggplot2, yet the basic function you begin with is just ggplot without 2. Then the first argument to the ggplot function are the data that you are going to be using. And note that I'm trying to maintain the same color code here, data, that okay, here between these different elements and the actual code and syntax. The next element of the ggplot function is aesthetics. And for aesthetics, we're gonna use AES. All right. So for example, the most common aesthetics are going to be adding and using the AES function are the X and the Y values. So what aspects of the data map to the X and the Y coordinates of this plot? But then to add function to this, to add element to this, we are going just to use this plus sign here and add a new row, and we are going to add the geometry to the plot. And this geometry, then here, after this hyphen, we can use whatever we want. It depends if we're going to use a scatter, a heat map, or whatever. So, but for the sake of this example, we're just going to create a scatter plot, and that is done by using the geom point. So geom point, generally, that means the graphic that I'm going to generate is going to be a scatter plot, and geom underscore point function. And within that geometry, we can add other aesthetic elements. So again, using AS function or aesthetic function, and now we can map out different values from the data to things like the color of the point, of the feel of the point, or any other aesthetic property that may be connected to the data. And remember that because these points, these different elements are connected to different aspects of the data, we literally make that connection, we make that mapping by just saying equals and whatever the name of the variables or the column in that data that you want to refer to. So in the original example, we had variable 1 goes to the x-axis, variable 2 goes to the y-axis, and then within join points, variable 3 goes to the color of the point. And finally, I'm sorry, and finally we can add another plus and another whatever other element we would like to assign, like coordinates, as I said, or team. But don't worry if this is a bit confusing. With time, you're gonna find your way, and we're gonna use it in the software. So you will see it's just easy to run and to plot data and generate whatever type of data we want. So I'm just gonna go back to the software now and to plot our data. So to plot our data, as I said, so the first thing I'm just gonna call the ggplot. That means I need to plot a graphic. And in that ggplot, I'm gonna plot my data here. So I'm just gonna use my data. Then after my data, I'm just, as I said, we're gonna use aesthetics. And for aesthetics, we're gonna use the AES function. And in the AES function, I'm just gonna assign the x-axis to the chromosome. So x is gonna be equal chromosome. And then I'm gonna, with the type, I'm gonna fill these chromosomes. So each chromosome, I'm gonna find different type of nucleotides. So I needed to each type of nucleotide to appear on this chromosome in a different color. So I'm just gonna do fill equal type. Okay, I'm sorry. I'm just gonna go a little bit up here so you can see what I'm doing. Then once I done that, I'm just gonna add what type of graphic I want. And once I want, for me, like when working at chromosome, I prefer to do my graphics as a graph and then just look to type of sequences on those nucleotides on my chromosome as different colors and different type. So I'm just gonna do plus geom for geometric presentation, and bar graph. To do it as a bar, I'm just gonna do it as a bar graph. And I'm gonna generate this to all my data. So I'm not gonna put any limitation. If I leave these brackets open, that means I'm opening all the data. And then I will just do a plot. Here, it said that there is an error. So I think I did miss a one ggplot error. ggplot made data. And okay. Oh, I used a small hyphen. I needed to use a big one here. So actually to call again, you just have to go with the up and down buttons to call the same function. So actually here, I just miss here. All right, now I hope it will work. All right. So here, what it told me, that the function ggplot, it doesn't exist on my laptop. That means this function, it doesn't exist. So if there is a function, it doesn't exist, I need to install that function. And as I said, the command to install a package, it just install always packages. Then I can go and look for the packages I want to install. In that case, I'm just gonna install ggplot2 here. Then enter, and it's just gonna install that one for me. All right, I'm just gonna look for it here to just see if it's indeed being installed. Now it is here. And now I think I can run my function nicely. I hope so. No, some function. Okay, it's still not working. So what it says here. Studio combine tools, sorry, package and C user update library. Speaking contribute in Windows. So actually I have just some. Once you don't have it, that means all the packages I have to look for it and zip it. So I'm just, once you are in that situation, here you're gonna learn how to upload new packages to your laptop. So usually when you are messing with one function or the laptops or the R software and not executing a function, usually you're gonna either propose to you a package that you're gonna look for it and in the format. So we can just gonna copy the link and we can just go and look for that link and download it directly. We can zip the file. All right, now the ggplot file is being downloaded. Now I just can go to the R, all right, and then ask him that to install the new folder that's been downloaded. So to install the new folder, I'm just gonna go and add file or open file directly, and from here, and this is the ggplot that he's been looking for. All right, now he just gonna open it. And we can use it directly. Now we can use this function again. Oh, install package file. I'm just gonna browse the package. I already installed. It's ggplot. I'm just gonna open this package and install it in my laptop. Now it's running it. Directory.
Walid, just a quick reminder, we have 7 minutes left
Really?
before the webinar ends.
All right, so I'm just gonna go directly and show you how it's worked. So usually, as I said, working with R, it's the easiest way, possible way you can work with. It's easy. And also it works with artificial intelligence as fine. Here, I'm just gonna show you results of our physiological data. So generally, when you are working with like data from BMC bioinformatics and mapping, actually we can work with complex data. And using R, using the crane, using the chromomap, we can do all these things. That means selecting the gene, analyzing the gene, and structuring them. And for that, actually, I encourage you to visit this article. I'm just gonna go for it. That gives all the scripts that actually they are needed to analyze data, genomic data, or meta-genomic data. So for the Chrome map, actually, it will show you how easy to use the R software. And actually, all the mapping you saw, we've done actually we've done it with Chrome map, which is another package that is easily used in the R studio. So you can map all your genes, you can do all the normalization you need. You can look for all the signaling pathways you need and make all the correlation. So usually when you're working with that, you don't have to come with your own script. Usually we just use like a pre-prepared script, like this script here, to do the mapping of the whole genome. And then this script, we can use it with online data. So we can go to a baseline data like NCBA, and we can do all the creating of libraries or the mapping or the comparisons to look if there is either a change in the gene, there is mutation, there is a problem with the reading. All that we can do it just with the R software, and we can generate all these kind of gene mapping, things that we cannot do with the classical softwares. Also, for people that they are doing electrophysiology, I'm just gonna go quickly. For people that they are working on electrophysiology, there is a number of packages that we can use. So generally, if we are working with EEGs or working with patch clamp analysis, the problem is the amount of data that they are generated. So using these packages on R like Reader, it will allow you to read through all your data. Then you can use Doppler Insider to clean and process all the data. And you can use TS outliers to eliminate the background noise or whatever you want. Then you can use spike R to look for spike amplitude and calculate for the frequencies. Also, you can calculate firing rates and creating pre-stimulus time histogram, PTSH, with the people familiar with electrophysiology by applying statistical models such as generalized linear model, JLN, or Bayesian model, or to analyze spectrum data mass. So generally, when using RStudio, you don't need actually an experience with programming, because most of the scripts you're gonna be doing actually it exists in packages, and it's easy to look for these packages on the CRAN website. Also, you can use the artificial intelligence. You can use ChatGPT just to ask which package to work with each system, and it just gonna guide you directly to it. Also, you can work with you can work with advanced models and do whatever type of analysis you want on a massive data, what we call big data or whole genome sequencing data or electrophysiology big data. So all that you can just analyze easily with the RStudio. And then you can create libraries. One friend of me asked me one question. When you are publishing with RStudio, how we can verify if your data have been properly statistically analyzed? So usually when we are publishing like electrophysiological data analysis, what you're gonna see on the paper is just the graphs. But with those graphs, actually, what we add as a supplementary, actually, it's the R file. In this R file, actually, we're gonna find all the details on the statistical test that's being conducted on this data to know if there is a significant differences, if there is a significant correlation. It will inform you about the type of test, the statistical test that's being used, if it's a correlation with person, if it's a sample regression, or whatever. So generally, it's easy to work with the RStudio. And now RStudio actually it adopted another function in the realm of the machine learning in big data. R added a new packages like Carat, Random Forest, and XBlot. And these packages actually it work with the machine learning in big data analytics. So for people working like for in psychology, and they are working on people behavior, I don't know, on drug addiction or buying, or they're gonna work with massive data that they can generate through, I don't know, internet or social media. So they can still use R because it has the ability actually to analyze these data and to do what we call a decisional tree to show you the tendencies, to show you the solutions. So in conclusion, I'm saying that actually R is good. It might work. It's flexible. You can do with it whatever you want. It's not stagnant. It's not like the old software. You have already fixed script and embedded in the system that you cannot modify, and you have just to be strict of the function afforded by the softwares like SPSS or GraphPath or whatever. So the transition for this classic statistical software to RStudio is not merely a trend, but a necessary evolution in the practice of modern data analysis. So the flexibility of R in its cost effectiveness, community support, because people each time they are developing script and putting them online for free, and its reproducibility and integration with other tools, it makes it indispensable asset for contemporary statisticians and people doing research. So embracing RStudio means like aligning with the future of statistical computing, ensuring that one's analytical capabilities remain the cutting edge of technology and methodologies. And before ending, I will just encourage you to go and look for these online tutorials. It's gonna help. And once you are going to, you're gonna find like it's really easy to work with our software. It's not complicated, even though it seems like it's a programming, but it uses a simple language. It's very intuitive. It's friendly user. It doesn't take time to learn the R. And once you're gonna find your way with it, you can do, as I said, the limit with R in analyzing data and presenting data is the imagination. So with that, I said thank you very much. And sorry if I depassed my time.
Thank you, Walid. Thank you very much for this very interesting and exciting presentation. Without wasting time, I move directly, if Lile agrees, to the questions.
Yes, we definitely can go to questions, Neza, but I think maybe we will limit our questions to only 3, with a proposition that we have a workshop on statistics during the SONA meeting, which is in 2025. The symposium, what do you call them, call is open, and I would like Dr. Abosi, maybe with Neza and myself, to connect after this and talk about a workshop that we can have during SONA, either a pre or post workshop. I think this will be excellent because we really need more of this level of information. Walid, thank you so much. You can continue, Neza, and then at the end you can close for us. This is my last comment for the session. Thank you.
Thank you, Lile. Okay, it's an excellent idea. We can definitely work on that together to prepare it. So
Yes.
I start with Lumbuka Konda questions, who asked how do you input data from an Excel into R? I think that question was already answered during the presentation.
Yeah, it's easy. Actually, once you are in R, actually there is many ways. Either you go to the environment and you have a button to input data sets. But when working with R, the best thing to do actually is to convert your Excel data to CSV data. So especially if you are working with massive data. So because R is going to be using the memory of your laptop. So if the memory it's basic, I prefer that you're just going to transform all your data to CSV and then work with it easily on R than using it as Excel file. But it works with Excel file. It's just going to be lengthy to execute all the function that they are asked from R to do it.
Okay, thank you. And the second question from Gladys Ngupay. Thank you for this great presentation. Please, could we have the bioinformatic link mentioned in your presentation?
Yeah, of course. Actually, I can send you all the presentation, then with all the links to install it, to use it, and with all the tutorial links to learn how to do it. It's easy, as I said, and it's a friendly user. Yeah, I can do that. Yeah, of course.
Okay, thank you so much, Walid. Is there any other questions from the audience? If not, I have like a kind of stupid question.
There's not stupid questions at all.
So the question is, you mentioned that R is more suitable for massive data.
Yeah.
And usually in neuroscience, especially when we are working on behavioral neuroscience, we do experiments with reduced effective on animals. Is it necessarily to go to R? I know it's the tendency to go to the programming softwares, but if we have the choice, is it better to stay with this, with the conventional, let's say, softwares, or we should do this transition?
Okay, so learning R, I think, is a must, because with time you're gonna find out that you're gonna be in a project that imply a lot of data. But where you are working in an experiment with limited output, like it's like we are not talking about millions of outputs. We are just talking about hundreds or whatever outputs. With that, actually, SPSS of GraphPad Prism are the best. Even now with GraphPad Prism, I was looking to the latest version, like for the graphical like presentation that was lacking from these old softwares. Actually, it became very nice and very very good to see and to deal with, and it became easier. But SPSS, yeah, it's good. It's gonna do all the work, but you don't need a tank to kill a fly. So yeah, so I encourage to use all of them in it's adapting, because with the other softwares, it's easy to work with. It's just question how you're gonna organize your data. The advantage with RStudio is it's no matter how you organize your data, because you can put whatever data you want, and then you can tell him how you want it to be, and he gonna just do it for you. That thing you cannot do with the other software. But if it's small data and you can prepare it nicely and clean, yeah, you go with GraphPad Prism or SPSS or SAS, whatever software you're using.
Okay, thank you. I see a hand raised, Temeka Chika Igwe. Don't know if you have the possibility to speak, to activate your microphone. Otherwise, just write your question on the question and answer session.
Okay. Oh, okay. Can you hear me?
Yes.
Yeah.
Okay. Thank you for Doctor another person, other senior colleague that anchored this program or this training. This is my second time of hearing about R. The first time I heard about it, I ignored it. But from what you have presented, it seems that I have to change from SPSS, or at least know also a little bit of this. But let me just go straight to what I want. I don't know whether somebody have asked about it or you have commented about it. Please, is it possible for us to have this video in our mails?
Yeah.
No, okay. Thank you so much. Thank you so much. That's just what I wanted to ask. Thank you.
Yeah. All right, you're welcome.
So thank you so much. If there are no more questions, we will
Sorry, Neza.
Yeah, sorry.
I just want to add to the gentleman who was asking if the information can be found in their mail. Tandega will post everything that Walid has given us on the SONA website. You can go to the SONA website. Should you not be able to navigate the SONA website, then you can send an email to the inquiries in the SONA website, and this will be sent to you. Sorry, Neza, you can continue.
Yes, and just to mention that all the webinars are already on the SONA website. So thank you so much. Thank you, Walid, for again for this presentation. Thank you to everybody for your attendance, and hopefully we can meet all together in the 5th SONA webinar next month. Thank you so much.
Thank you.
Have a nice evening,
Bye-bye.
everybody.
You too. Bye.
Bye.
Bye.